without further ado, I'd like to introduce you to the topic of today's presentation, which is biochar as a soil amendment, the reduction of mercury transport from hydraulic mine debris. So the focus of this project is on hydraulic mine debris and the impact hydraulic mining continues to have on water quality. Hydraulic mine debris is defined as alluvium that was displaced by hydraulic mining and continues to discharge some sediment into streams and rivers of the Sierra Nevada during storm events. Remediation of heavily mined watersheds such as the Yuba River are critical to improving water quality, and water quality can be defined as the measure of the condition of water as it relates to biotic needs. Hydraulic mining that occurred in the Sierra Nevada continues to degrade water quality, especially in the Yuba River watershed. So a bit of history. Hydraulic mine debris discharge into the Yuba River led to downstream flooding of the town of Marysville. Edward Woodruff, pictured on the right here, is a member of the town brought against the North Bloomfield Mining Company for debris discharged in 1882. This case was presented to Judge Sawyer in 1884, which led to the Sawyer decision, which restricted hydraulic mine debris from being discharged into navigable waters. However, in 1893, Congress passed the Caminetti Act, which allowed hydraulic mining to begin again under the conditions that a license be obtained and debris be prevented from entering navigable waters. And it's estimated that in California alone, there are 40,000 abandoned mine features which continue to degrade water quality. So hydraulic mine debris continues to impact water quality and increase suspended sediments and heavy metal concentrations. Hydraulic mining used water cannons, pictured in this image, to extract the placer gold deposits from the Sierra Nevada. And sluice boxes captured the sediment slurry and separated gold from gravel particles. So to increase the mining yield from this method, mercury was added to these sluice boxes. And gold and mercury have a high binding affinity and create an amalgam with a higher density. The downside to this is the sluice boxes lost on average between 10 and 30% of mercury to the environment annually, based on work by Churchill in 1999. So as a result of this, mercury continues to be discharged from hydraulic mine sites during storm events. So mercury was mined in the Coast Range Mountains and then transported to the Sierra Nevada. These mercury mines are indicated in red on this map that was adapted from research by USGS. Hydraulic and hard rock mines that used mercury are indicated in gold, and it's estimated that 26 million pounds of mercury were brought to the Sierra Nevada based on calculations by Churchill in 1999. So mercury was mined in the Coast Range and imported to the Sierra Nevada. The oxidation of this mercury turns into mercury 2+, which can undergo biogeochemical transformations into methylmercury with the addition of a methyl group. Sulfur and iron-producing bacteria can facilitate this reaction as part of their metabolism in oxygen-limited environments. Mercury present in aquatic systems is then biologically available to these benthic microorganisms, which increases methylmercury production. Methylmercury is a form of mercury that is most readily incorporated into biota and most toxic to humans due to bioaccumulation and biomagnification. So bioaccumulation is the process by which heavy metals increase inside a single organism, and over time mercury can bioaccumulate to potentially toxic levels inside the single organism. Biomagnification is the process where mercury and methylmercury concentrations increase from water samples into biotic tissue on the order between 100 and 10,000 times from water samples into bacteria, based on research by USGS researchers, including Di Pasquale in 2009. As bacteria are consumed by secondary and tertiary predators like fish, these levels of mercury biomagnify. Consumption of tertiary fish species expose humans to methylmercury, and to reduce this risk, the remediation of mercury-contaminated sources is a priority for water quality and local communities. So this image is of the Humbug Creek South Yuba River confluence after a storm event. Suspended sediment and mercury are mobilized. This is harshly indicated by that orange cloudy color of the water entering the river. Hydraulic mine sites contribute mercury to watersheds during these storm events. The majority of mercury is particulate-bound mercury, which is defined as mercury bound to sediment less than 0.063 millimeters, known as fines. Particulate-bound mercury can stay in suspension for more than 18 hours, increasing the mobility of mercury away from contaminated sites, and this causes increased mercury contamination throughout watersheds. So mercury contamination of soil, water, and biota creates challenges for remediation. Some current strategies include removal, capping, and additions of soil amendments. Removal of mercury-contaminated soils can be very costly and causes resuspension of that particulate-bound mercury fraction, which further distributes pollutants and particulates from the site. Capping involves a topical application, typically of clays or limestone, to contain these types of pollutants. However, this method doesn't account for groundwater flow mobilizing contaminants beyond this capping layer. An alternative method uses a soil amendment called biochar incorporated into the soil, which has been shown to sorb mercury in field and lab trials. So what is biochar? Biochar is made of organic pyrolyzed material, a process using thermal decomposition to create chemical binding sites. Biochar has been shown to remediate both organic and heavy metals and can be made cost effectively. Incinerators and air curtain burners pictured on this slide can be brought to contaminated sites, and the use of native materials and reducing transportation costs. Recent field studies have support the application of biochar to remediate mercury-contaminated sites. Studies over 100 weeks have indicated that biochar has a mercury sorption potential capacity of greater than 80%, and in experimental lab columns, sediments containing biochar retain twice as much mercury as compared to control sediments. So how does it work? Recent research by Kilowite in 2011 has shown that biochar made of lignin promotes mercury absorption. So lignin is a compound found in trees, pictured on the right, and is composed of many aromatic groups. Lignin has the potential to become the functional groups present on the bottom right when biochar is created, which are crucial for the mercury absorption capacity. Biochar pyrolysis temperature and organic material determine the binding sites that are present. So these criteria allowed for sorption species to be selected, in our case, specifically mercury. So to create a biochar with a high sorption capacity for mercury, low pyrolysis temperature between 300 and 400 degrees Celsius, combined with an organic material high in lignin, creates a more effective biochar compared to other temperatures or other feedstocks. Under low pyrolysis temperatures, increased concentrations of the aromatic compounds are produced, and these compounds contain negatively charged functional groups, which have increased sorption capacity for heavy metals like mercury. Organic material selected also contributes to the potential functional groups present. Pyrolysis of lignin produces higher concentrations of ketones, aldehydes, and carboxylic acids as compared to grass-derived biochars. So based on chemical binding affinity, biochar manufactured using ponderosa pine at 400 degrees Celsius was selected for experimentation. So the focus of this study and thesis presentation is to present the results of the effect of biochar on stormwater runoff and hydraulic mine debris. Laboratory experiments were used to determine if biochar improved water quality, and these experiments can hopefully inform future field studies on application rate and method. These thesis questions were used to evaluate the efficacy of biochar application. So 1, what is the effect of biochar on turbidity and mercury concentrations from stormwater samples collected at Malakoff Diggins? 2, what is the effect of biochar on turbidity and mercury concentrations from hydraulic mine debris runoff created using soil columns? And 3, what is the effect of biochar on turbidity and mercury concentrations in simulated rainfall runoff tests using hydraulic mine debris mesocosms? So to introduce you to the 2 sample sites, this is Malakoff Diggins and was once the largest hydraulic mine debris in California. It's now a state park and is located 26 miles northeast of Nevada City and is approximately 3,200 acres. The site geology consists of Eocene auriferous sediments, which are subject to rill and gully erosion where debris flow occurs. So this is an image of Malacoptigans today, and today Malacoptigans is drained by Hiller Tunnel into Humbug Creek. Malacoptigans contributes 100 grams of particulate-bound mercury to Humbug Creek annually. Research done in 2011 by USGS conducted dredge tests on hydraulic mine debris at the Humbug Creek confluence of the South Yuba River and resulted in total mercury concentrations greater than 100,000 nanograms per liter. So this is the second mine site that was selected. It's located downstream from Malakoff on the South Yuba River near Smartsville, California. This mine site is much smaller. It's only 520 acres with different site geology consisting of basalt bedrock overlaid by auriferous gravel. Both mine sites were mined similarly with water monitors and sluice boxes lined with mercury, which left contaminated hydraulic mine tailings throughout each site. This is Blue Point today. Blue Point was also drained using a tunnel similar to Malakoff. However, fewer samples have been collected here as compared to Malacoptigans. Concentrations and loads were measured on February of 2019 to determine the potential contamination rate. Stormwater was collected from the tunnel present at Blue Point Mine, and stormwaters were registering at 70.1 nanograms per liter with an instantaneous load calculated at 5.8 nanograms per liter. So 3 laboratory experiments were conducted using soil columns containing hydraulic mine debris, columns containing hydraulic mine debris and mesocosm trays to determine the effect of biochar on turbidity and mercury concentration. Turbidity was assessed using a Hanna turbidimeter, and experimental samples were collected in acid-washed 125 ml sample bottles supplied by Brooks Applied Labs. An analysis for mercury species was performed according to EPA method 1631. So our first experiment, stormwater columns. Stormwater samples were collected from Hiller Tunnel in 1 liter HDPE bottles in March of 2019 following a storm event. Experiments were performed using the PVC columns you see on the right. These columns measured 30 centimeters in height by 5 centimeters in diameter and were secured at the bottom using 20 mesh to retain the decomposed granite. Discharge was passed through columns containing a layer of decomposed granite as packing material, overlaid with a lens of biochar. 0, 2%, or 5% biochar additions were analyzed for this effect on turbidity, total, and dissolved mercury concentrations in 3 replicates over 24 hours. Okay, sediment sample columns. These columns that were previously described were then refilled with biochar and 200 grams of hydraulic mine debris collected from Blue Point Mine. Biochar and hydraulic mine debris were incorporated together using a steel scupula at rates of 0, 2, or 5% additions, and then DI water was passed through each column and captured in the 1 liter glass beakers that you see. Flow-through was then analyzed for turbidity, total, and dissolved mercury in 4 replicates over 24 hours. And sediment mesocosms. So sediment mesocosms were performed in these trays containing 6.5 liters of sediment collected from Blue Point Mine. 2%, 0%, 2%, or 5% biochar was incorporated into the sediment in each of these 5 replicates, and 3 liters of DI water was applied to simulate rainfall, and the trays were placed on a 10-degree incline to promote flow out of the spout in the front of the tray. The outflow was collected in 1 liter glass beakers, and flow-through was analyzed for turbidity, total, and dissolved mercury. The grain size distribution. Grain size distribution for the sediment collected at Blue Point Mine was analyzed using a Humboldt shaker model for 10 minutes. 3 replicates of 500 grams were passed through sieves ranging from 4 millimeters to 63 microns to determine the average grain size. And the grain size distribution analysis will determine what % of the sediment sample from Blue Point Mine is classified as fines and can be potentially related to mercury. So to introduce you all to some of our findings, these graphs show the effect of biochar applications on total and dissolved mercury. The graph in the left in blue indicates the changes in total mercury present in stormwater samples with and without the applications of biochar. After 5% biochar additions, the total mercury decreased based on calculated averages indicated with that thick black line in each plot. The graph on the right in green shows the changes in dissolved mercury fractions within stormwater samples with and without the different biochar applications. And after 5% addition, we can observe the greatest decrease, but not significantly greater than 2%. All right. In the sediment column experiments, biochar reduced both total and dissolved mercury. The left side graph indicates the biochar additions of 2% and 5% significantly reduced total mercury. However, 5% additions were not significantly greater than 2%, and the graph on the right shows the change in dissolved mercury present. Biochar also decreased dissolved mercury concentrations, but these results were not significant. And sediment mesocosms. The addition of biochar to sediment mesocosm trays significantly reduced dissolved mercury, but increased total mercury and particulate-bound mercury, along with turbidity. The graph of total mercury at the left shows a significant increase when 5% biochar was applied, and the graph on the right displays the results of the dissolved mercury analysis and shows a significant decrease of dissolved mercury present in the sample. So if you don't mind, I would like to take a moment to focus on the graph that indicates the total mercury concentrations. This increase in total mercury when biochar applied was very unexpected. The previous two experimental results showed a decrease in total mercury and dissolved mercury after biochar was applied, and this next graph may help to determine the reason behind these unusual findings. So this graph shows the relationship between turbidity and total mercury under the different biochar applications. The solid blue line present represents the control mesocosms without any biochar, while the dashed dot line represents mesocosms containing 5% biochar. The dot dash line showing 5% biochar additions indicates that as biochar application increased, so did the concentration of total mercury and turbidity present in each sample collected. The application of 2% biochar did not have these effects, as indicated by the relative closeness of the solid line and the dot line in each trend. The addition of 5% biochar increased turbidity and total mercury significantly. This may reflect biochar application and the sample collection method. Sediment samples from Blue Point Mine were then analyzed for grain size distribution. And of the sediment, between 8 and 12% of the sample falls below the 0.063 millimeter cutoff, which is under the classification of fines, clans, and says. This portion of the sediment in the sample from Blue Point Mine can be associated with mercury based on research by USGS and Fleck et al. In 2011, which observed that the highest concentration of mercury is related to sediment samples under this size fraction. So to conclude this research, the previously stated research questions can be addressed and then hopefully answered. So 1, the addition of biochar reduced turbidity and mercury concentrations from stormwater samples. 2, the total mercury from mine debris runoff. 3, the addition of biochar reduced dissolved mercury concentrations from mesocosms, but increased turbidity and total mercury in experiments with 5% biochar. So to discuss this, the addition of biochar to hydraulic mine debris and discharge reduces both turbidity and mercury concentrations. Increases in turbidity and total mercury concentrations in mesocosms may be due to the method of application on a larger scale. Decreases in the dissolved mercury concentrations may be attributed to chemical binding sites based on research from Kilowhite in 2011. Research by the Smithsonian Institute and Gilmore et al. In 2018 applied biochar to freshwater environments, and they saw a reduction in methylmercury concentrations in biotic tissue by up to 90%. So some future research on the methods of application is needed to in order to determine the best field application for biochar to contaminated sites. Retention structures such as wattles may prove effective at withholding biochar in place in eroded landscapes, and may be a potential method that could be studied. I would like to thank everyone that joined us today, specifically the dedicated faculty that have helped me get to this place: Dr. Carrie Monahan, Dr. Dave Brown, Dr. Jackson Webster, and Dr. Robin Donatello, and to my cohort, my family, my friends, and fieldwork partners. Thank you all.
If you would take a moment and unmute yourself, and let's give Madison a round of applause. Thank you so much, Madison. That was very well done. We'd like to open it up now to questions. Maddie, maybe keep sharing the screen in case going to any of the slides is helpful. If
Sure.
you would like to submit a question using the chat feature, this would be a good time to do so. Anyone in the audience is welcome to submit your question. At this point,